For the first one, we have, and again, these are all listed in 5' to 3', UUC to CUC. What does UUC encode? For those that did this particular problem, if you can remember. Do I need to throw up the codon code for us as well? All right, there's our codon code. What's UUC? PHE, phenylalanine. And then CUC? Leucine. Okay. So, anybody know what these, well, what kind of mutation is this? Given just that information there, what kind of mutation is this? And the options are missense, nonsense, and silent. Missense. Perfect. All right. So I might as well just draw the structure of these side chains here. So leucines, or if this is our amino acid, that's our basic structure. Phenylalanine looks like this. And then leucine looks roughly like this. Similar, different? If different, how different from one another? What would you say? And who answered this one? Devin, what did you say?
If it changes the protein structure, or the protein.
Or if it changes the function, I guess.
I said yes, just because I said that they were different amino acids.
Yeah, and typically that would be a rule. That would be a rule of thumb that we totally agree with. That yeah, if there's a change in the amino acid, yes, it's going to change the structure of the enzyme. Sometimes we find that some of these mutations, though, don't completely change the function. And that's where I was trying to prelude this on Friday by saying we have to sometimes actually take a look at the amino acid itself to determine if there is an effect. Now, we would describe this one as being bulky. This is a pretty large, it's pretty flat as well. It takes up a lot of space. Whereas this one from leucine, fewer carbons, we might say that because of the size difference of these side chains, that would affect the structure and the function of the protein. Okay? There are examples in which this may not occur, though. So let's take a look at that. Let's go to C real quick. AUU to GUU. So what two amino acids are encoded by AUU and GUU? ILE and then val. So isoleucine and valine. And what I'm going to do is I'm going to draw the structure of these two real quick. So there's the structure of those two. What do they have in common? What properties do they have in common?
They have almost the same hydrocarbon tail.
Yeah, they have almost the same hydrocarbon tail. The only addition is this extra CH3 group or this extra carbon off of the isoleucine. What's true about hydrocarbons? Polarity. Polar, nonpolar? Nonpolar. So these would both be considered to be nonpolar amino acids. They have a similarity in structure. There's not too much difference in similarity. Even though this, again, is another representation of a missense mutation from isoleucine to valine, So you might get two opposing answers here. One person might say, well, it's changed the amino acid composition. Of course it's going to change the function of the enzyme. Somebody else might say, yeah, but the two of them still have some similarity in their properties. And so though it may change some of the function, it's not going to change, it's not going to completely delete the function of the enzyme. It's still going to be able to perform some of that function, maybe just not as well. Okay? So that's why, even looking beyond whether it's a silent or a missense or a nonsense mutation, sometimes we actually have to look at the amino acid change itself to determine, is there an effect? And if so, then we try to figure out what is that effect going to be. One of these up here represented a nonsense mutation. Which of those represented a nonsense mutation? Which one was that? Sorry, D, you said? So what's the hallmark of a nonsense mutation, Dana?
That encodes for a stop codon
Good.
rather than encoding for a different amino acid.
Good. So it starts out with a different amino acid, in this case UAU, which is, I don't remember, is that another phenylalanine? Tyrosine? Okay. Yeah, okay. And then UAG here would be one of those three potential stop codons that occurs. Does this have an effect on protein function? Absolutely. Usually if this is occurring anywhere within the active site, it disrupts the active site. It also leads to an early stop in the translation. Therefore, any of the amino acids that are occurring after where this mutation has occurred won't ever be translated. And so these have quite deleterious effects or quite negative effects on proteins. Which have the least amount of effect?
The silent.
The silent. Why, Derek?
Because typically when the silent mutation occurs, it keeps the same amino acid, so it doesn't change the function.
Yeah, and that's because there's redundancy built into this code again. And I somehow didn't include one, so this down here, UUA, was B1?
These is kind of-
Yeah.
Oh, well, I didn't pay attention to my own code then. UGU to UGC, cysteine. Okay, perfect. So that one would be an example of a silent mutation. Again, there's a lot of redundancy built into the code. We won't exactly talk here about why that redundancy exists, but that redundancy does have some benefit here in that not all mutations are necessarily going to be ones that change the structure and function of a protein. So let me pause here then and see, any questions on this? It seems like you guys have a good grasp of just the types of mutations. Any questions? Stop it.
So when you were saying to kind of go beyond and look at the amino acid,
Yeah.
how would we want to look that up or
Yeah, so that would be something where I would say, if I were to ask a question like that, I would actually give you the structure of the amino acids.
Okay.
And usually what we're paying attention to is what's on the side chain. So is it hydrophobic like these are, or does it have a hydroxyl group in both cases? There are examples where mutations don't have an effect. But if it does drastically change the structure or change that side chain drastically, then you can say, yep, has an effect. I just wanted to point this out as an example of oftentimes we immediately think if there's a missense mutation that does lead to changing the structure and function of a protein. Not always true. Sometimes we can't detect the change in the function. Okay? And sometimes that depends on where that mutation even takes place too. But that is not something I would ask you to know. I have my cheat sheet of the amino acids on here because I once upon a time had to memorize every one of these and now know none of them anymore. So, biochemistry, don't take it unless you want to. All right. Other questions? Okay, so let's change focus briefly. How do mutations occur? In what ways can mutations within, and let's focus here, we'll just put it in the context of bacterial cells. How can mutations occur? There are a couple of different reasons or methods. Anybody have an example? Ashley.
Ionization?
Yeah, ionizing radiation. So, or even just UV radiation as well. Ionizing radiation we talked about is a great form of controlling growth. And what it does is it creates double-stranded breaks in the DNA. In other words, it causes breaks in both of those backbones. And what bacterial cells have to do is they have to try to piece that back together like a puzzle. But it's a puzzle where all the ends are flat. You don't have nice little grooves to help guide you where anything goes. And so oftentimes what happens is these puzzles get put back together incorrectly. And that's what can lead to the death of the cell if enough ionizing radiation has occurred. UV, on the other hand, creates thymine dimers. And so these are cases in which we have two thymine nucleotides next to each other in a DNA sequence. And what happens is those thymines actually, a covalent bond gets formed in between those nitrogenous bases. So what it ends up creating in the DNA is essentially kind of like a little pucker where those have bonded together, but they're still base pairing across with the alanines on the other side. That pucker can get misread when the DNA polymerase comes through to read that sequence. And so that can create either point mutations or insertions and deletions as well. Okay. Other examples. Those are kind of the two major ones. There are always going to be chemical carcinogens, or what we classify as carcinogens. Their effect is on DNA structure as well. So occasionally we can get, and this goes beyond really what we're concerned with, but there are times in which mutations can occur where, if I'm not mistaken, it's cytosine, an amino group gets removed from the cytosine, turns it into uracil. And so instead of a CG base pair, you accidentally get a UG base pair. And the cell tries to fix that. Well, the U gets replaced with a T in the DNA world. And so that creates a TG mismatch that would create a mutation. Those are related to carcinogens, more often what we would talk about with eukaryotic DNA. The last one is completely random. Mutations can be a random thing. We talked briefly about the DNA polymerase, and one of the things we said about the polymerase is that it makes mistakes, but those mistakes are rather rare. Maybe making a mistake one every, I don't know if I have this on here or not. Yeah, here we go. So basic information, roughly there's about one base pair mutated for every 10 to the 9th or 10 to the 10th that are replicated. The DNA polymerase has the ability to proofread. So if it realizes it's put down the wrong nucleotide, it has the ability to back up, take out that nucleotide, and put the right one so that there's a proper base pair match. But this isn't perfect. There are mistakes made. And eukaryotes and prokaryotes have repair mechanisms to recognize these mismatches. They're known as base excision repair, nucleotide excision repair is sometimes the word that's used as well. But we get about one mutation in every 220 to 2200 generations of a bacterial cell. In other words, the cultures that we grow up in lab, we've got somewhere on the order of 10 to the 9th cells, 10 to the 10th per milliliter. They're not identical as we oftentimes think about. There's actual genetic diversity in there. We can't always tease apart what that genetic diversity is. Okay? So some of these mutations can be just random. That randomness is what we think actually contributes to quite a bit of antibiotic resistance. And we'll talk about this a little bit more in the next couple of weeks, more about this theory, but random mutations in genes that are targeted by antibiotics can select for certain mutations. And we'll take a look at one example today, that being gyrase. Okay? All right. Give me a second of all the microorganisms in that population. Okay. Sometimes, though, maybe one cell is able to survive or to outgrow that particular minimal inhibitory concentration. That's the bottleneck. And what we get from this are then cells that are more resistant than others to that concentration of antibiotic, or a single cell that's more resistant. So in this model here, what this one is saying is if a bacterial cell has a spontaneous mutation that allows it to be or renders it resistant to an antibiotic, by adding an antibiotic, an environmental pressure, kills off all of the sensitive cells, allows those that are resistant to survive, and then now with this freed up space, they're allowed to multiply, they're allowed to utilize the energy source, and they become the predominant species or the predominant strain within that culture. Everybody following along so far? Any questions on that model? Okay. So let's go back to gyrase. What's the function of gyrase? Two roles. Prevent supercoiling, that's one. What's this bottom one represent? This bottom model here.
Separating the replicated.
Yeah, so separating those replicated chromosomes after the process of DNA replication. Gyrase is a molecule or enzyme that is inhibited by the fluoroquinolones and quinolones. And one of the things that we've said is that in cases where we treat with one of these types of antibiotics, we pointed out we can get these, what look like or have been described as snakes or filaments of bacterial cells. So for example, if we treat with the quinolone nalidixic acid, we go from cells that are normally rod-shaped with a chromosome in the middle at the nucleoid to now having these really long cells. They've continued to grow and elongate, but those two chromosomes can't separate from each other, and so the cell can't complete binary fission. That's what this class of antibiotics does to the cells. Okay. Specifically, I'm just going to point this out real quick. One of the things that we talked about with the gyrase is that its role to separate these two chromosomes, that actually gets blocked, and the mechanism by which it does is some of these drugs like ciprofloxacin and these other quinolones. Here the light blue is representing those DNA strands. This is the other one that's trying to move through. Ciprofloxacin blocks gyrase in this conformation right here. So gyrase is able to actually begin the process of separating those two strands from each other, but it gets stalled in the process. Okay? And so then the cell ends up not finishing replication, still is able to metabolize, ultimately dies though because it's not able to continue to reproduce. So here's what's been observed. From 1990 to 2005, this is data straight from the CDC, when it was still reputable. Now all governmental agencies are no longer reputable, apparently. So this is looking at percent of Neisseria gonorrhea isolates from hospital settings that were resistant or considered intermediate resistant to ciprofloxacin. Given that we're looking at resistance or intermediate, this would be based off of Kirby-Bauer assays. Okay? And so what they find is starting in 1990, a low percentage of them are found to be intermediate resistant to ciprofloxacin. And what happens over time? Yeah, not only does the intermediate percentage remains relatively the same, but that resistant population begins to greatly increase. In other words, by 2005, roughly 1 out of every 10 isolates or strains of Neisseria gonorrhea that were identified were resistant to ciprofloxacin, which was an incredibly useful drug against that bacteria. Here's how some folks think it happened. So this is data from a paper in which they were looking at individual isolates of Neisseria gonorrhea and trying to determine, well, what's different? If they're resistant, that must suggest that there is a change in the gene, some sort of mutation that has led to a loss, I guess you could call it a gain of function, which would be resistance to ciprofloxacin. How did that occur? And these are the different strains that they're looking at. And you can see here their resistance to ciprofloxacin, which is a quinolone. So these are minimal inhibitory concentration experiments. Nalidixic acid, which is another quinolone. And then novobiosin and penicillin, which are aminoglycoside and beta-lactam. So targeting something completely different. And what you see here is the cells are relatively equally sensitive to these two, but what we find is from one strain to a next, there's this, they've ordered them an increasing resistance, or it takes more drug to inhibit the growth of these microbes. Okay, everybody still with me so far? So the question is, how does this happen? How does this particular event take place? What we're going to do is, I'm going to skip this slide for a couple of minutes, but I want to go ahead and I want to take a look at this on here. And I'm going to give you guys a few minutes to work over it first. So question 5 here, it reads, in the table below, each of these strains, and I've just listed 4 of them, is an isolate of Neisseria gonorrhea that has shown increased resistance to ciprofloxacin. The letters under, I guess I should know what this means yet. 83 to 104 here represents the amino acid in the sequence of gyrase A, which is one of the 2 parts that makes up the gyrase protein. The 83 through 104 just means the specific amino acid in each of those locations in the full-length gyrase A. And you're going to see there that at 91 and 95, they identify actually a couple of different changes have occurred. So for example, in FA19A, there's no difference in the amino acid sequence between gyrase A of the FA19 and the FA19A. However, in the B and the C, we now start to see amino acid changes. So going from an S, which is serine, to an F, which is phenylalanine, or from a D, which is aspartate, to an N, which is, I think it's asparagine, but I always have to double check that. No, glutamine. Glutamine. I have to double check that real quick. Have I written it down? No. What I want you to do is the following. We're actually going to, instead of working forward like we have done, we're going to work backwards. We're going to go from the amino acid sequence, and we're going to try to figure out what potential mutations, what potential point mutations could have happened to lead to that change. The way that we do this is we start with what are all the amino acids, or what are all the codons that can be encoded for serine. So for example, we've got 4 up here, and actually serine's encoded by potentially 6. The other 2 are located over here. So start by writing all those down. And then start next by writing down the codons that encode for phenylalanine. And see if you can identify 1 or 2 examples where a single nucleotide change would lead to a conversion or a missense mutation from serine to phenylalanine. Okay? That's where I want to start. So I'll give you guys a few minutes to work on this. If you get that, go ahead and move forward to the next one as well. I'm going to figure out what N is real quick, because I forgot. You know, Erica, we made a lot of plans for the honey badger plunge without you on Thursday, too. Well, that was the new name that got. You didn't inform her of the name change? You didn't inform her of the name change? Yeah, there's just so many things that we talked about. It really was a critical day. All right, let's take a look at this one. So again, all we're focused on this one is just, let's focus first on the serine to phenylalanine change, okay? So what codons encode for serine? We've got UCU, oh, booger. Oh, booger. UCU, UCC, UCA, and UCG, right? And then what else do we have? AGU and AGC, okay. And which ones, what are the codons for phenylalanine? UUC. Okay. So we're looking for a point mutation here. In other words, just a single nucleotide change that would lead to the conversion from a serine to a phenylalanine. In other words, the simplest way to do this is actually just to compare those two lists to each other and look for, well, what single change can I make that leads to this? So which ones did you identify? UUU and UUC, yep. Sorry, those two to... Okay. So if we started with UCC, which would be the wild type or the original codon sequence, and we went to UUC, that would be an example of such a point mutation. And again, what kind of mutation is this? This is our missense. We also could see UCU to UUU. Both of those have something in common. What is common between those two, other than that it changes from a serine to a phenylalanine? What specifically in the codon? Yeah, it's that second nucleotide, right? The C. And so doing this, we've actually just identified the specific amino acid or the specific nucleotide change that occurred in this species to allow for that conversion from a serine to a phenylalanine, and has also led to some resistance of this microorganism to ciprofloxacin. Okay. Using that same principle, then, we can identify the same sort of change from an aspartate to an asparagine as well. Okay. So let's do that one real quick, and then we'll finish up with this conversation. So what are the aspartate amino acids, or ASP? GAU and GAC. Are there more? Those are the only two? Okay. How about asparagine, ASN? AAU, AAC. So which nucleotide in this one? It's the first one. Regardless of which of those two codons was used, it's the first one. Okay. So again, we've identified another mutation that has occurred that in this case, most likely what this mutation has done is it's made it so that ciprofloxacin is less likely to bind to gyrase. So it's lost its inhibitory activity on this particular protein structure. One of the things that we don't focus on is oftentimes there's a trade-off here with this gain of function resistance to ciprofloxacin. There's usually a trade-off. And the trade-off here probably is the cells are a little bit slower at replication. Their division speed is probably, or generation time is probably a little bit slower. Would be a likely trade-off there. God, I'm like walking over everything this morning, sorry. bull in a china shop. Any questions on how to do this or how to identify these types of mutations? If you want practice problems with this, there's more in here, but also just to let you know, I did this kind of problem on the exams the past 2 years as well, so that you might find more practice in those as well. The story goes here with this particular example is that there's more than just these 2 mutations that have occurred. There is another molecule or another amino acid, or sorry, another protein. That's what I'm trying to say. Topoisomerase encoded by this PAR-C gene. What I didn't show you in this initial table is actually there's more than just mutations to gyrase. Ciprofloxacin and the quinolones also can inhibit topoisomerases. There's a lot of similarity in the function between them and the gyrase. And so what we find is that actually when we take a look at all of these strains that have shown resistance, B, C, D, and E, you can see they're put in order of increasing number of mutations that have occurred to one or the other or both of these particular proteins. Okay, and that seems to account for the resistance that they've observed to ciprofloxacin at least. Okay, and so the rest of this case study then just kind of walks you through going through these remaining parts and figuring out the rest of these mutations. I do want to point out then what does this mean? And again, the change here, that single nucleotide change, allows it so that the enzyme can still perform the majority of its function. It just inhibits its ability, in this case, to bind gyrase. And that's what I'm trying to model up here on this particular slide. So that it no longer allows for ciprofloxacin to bind to the active site. Any questions? Devin? Yeah.
How do you know that the specific strains are resistant to the antibiotic? Is it that it has the letters by it?
That's not telling you that it's resistant. It's actually the minimal inhibitory concentration. So if you take a look at this right here, this is the minimal inhibitory concentration of ciprofloxacin that it takes to inhibit the growth of these microbes. And one of the things that we said is if 2 microbes show, or if one microbe is more resistant than the other to an antibiotic, it will take more of that drug to inhibit its growth. Therefore, the MIC is going to be larger. So they have just put these in order of increasing concentrations of ciprofloxacin that it takes to inhibit the growth.
So like after the N by the 195, what does that mean?
So again, what that means here is just that it's telling us this is the protein sequence of gyrase A between those two amino acid 83 and 104 in the sequence. At 91, there's been a conversion from a serine to a phenylalanine in these strains here, and from an aspartate to an asparagine in these last 3 strains here. That's what it's trying to tell us. Okay? Any other questions? Okay, so what I'm going to do then is hand back exams. So, real quick, better than the first round. I always like to see that. Do you guys want to know the average? 81 was the average, and in comparison to the first exam, that's up 6 points. That's basically a half a letter grade right there. In comparison to the afternoon class, the averages were separated by 0.2 of a point. So there's really no difference in the 2 sections of the class on how you performed. I would say that everybody did really well, actually, for the most part, on multiple choice, some of the fill-in-the-blank type questions. It was the essays that tricked some people up, and admittedly, I think one of those might have been my fault, which was the one that I asked about, okay, so you're giving this chemical to somebody, what do you tell them? How to use it safely and effectively? And what I was looking for there was, one, it's a disinfectant. Make sure you wash up or make sure you wear gloves or something like that. And then secondly, for effectiveness, one of the things that we kind of went through a couple of times, or at least I repeated this phrase, is an effectiveness of a treatment is based on the magnitude or concentration, and also based on time of exposure. And so that's what I was looking for in that particular answer. Your ability to identify minimal inhibitory concentrations was really good. On the last question, most commonly the issue was folks didn't address the therapeutic index, which was the key part to determining which of these two antibiotics would you give to an individual. Almost everybody commented on the difference in the minimal inhibitory concentration. The toxic dose, which was in the table with those different antibiotics, was what was missing. Some kind of description of that. With the key, you now should be able to take a look at everything and identify mistakes. If you have questions, please come talk to me. And I feel like I need to comment on what homework is tonight, but I can't remember off the top of my head, so give me 1 second. Oh, I remember what it is. So for the homework tonight, on Wednesday, we're going to talk about methods of gene transfer. So bacterial cells not only pass genetic information from mother to daughter cells, that's vertical, but also we can see information being passed between different species. So methods known as conjugation, transformation, and transduction. In your group of 4, this is going to be a wiki one. I've given a set of terms. Each member of the group should choose vertical gene transfer or transduction or conjugation or transformation. Take those terms and put the terms that fit with your particular form of gene transfer. The terms can be used in more than one list, but this will just be a way to kind of start to get you to mentally organize this information. Just one due date for this one, tomorrow evening. All right? All right, once you get these, feel free to head out of here and have a great Monday.
We'll do that post-survey.
I'm sorry that there is.
We'll do that post-survey.